Hey everybody, I'm Byron Crump from Oregon State University, and I'm here to tell you a bit about the samples that you're using for your metagenomics toolkit workshop. Now, the data set you're going to be using today comes from the Congo River Basin, which is the second largest watershed on Earth and the largest watershed in Africa. Our colleagues Bienvenu Dinga, Paul Mann, and Max Holmes collected samples from sites in the Republic of Congo several years ago and published a series of papers about riverine dissolved organic matter, about greenhouse gases, and the general biogeochemistry of the Congo River Basin. The sampling sites for this project spanned a range of stream orders, from very small first-order streams all across the basin to the main stem Congo and Brazzaville at stream order 9. Now, these sites also span a range of landscape types, particularly in the amount of wetlands, which ranged from 0 to 99% of the watersheds. Mann et al. Found that river chemistry varied with landscape type, which appeared to influence dissolved organic matter concentration, dissolved organic matter chemistry, and greenhouse gas concentrations in the water. But they also found that chemistry varied with hydrologic regime, ranging from small streams with high flow to large rivers associated with each of the different landscape types. We used discharge estimates and watershed area to calculate the flow-weighted water travel time, that's this travel time here, for each sample. And this estimates the amount of time that water has been in the river channel at a given sampling point. So this tells us something about the average age of the water, and this time component is important from the perspective of microbes who require time to grow and adapt to the river environment. So our travel times, they ranged from less than a day in the smaller streams to almost a month in the Congo River main stem. So the biogeochemists found that the terrestrial landscape plays an important role in organic matter and greenhouse gas dynamics. So we asked this question: does landscape and hydrology structure river microbial communities? My student Ted Bambakidis used 16S ribosomal RNA gene amplicons to survey river microbiome diversity, and he produced this co-inertia plot that shows the relationships between microbial diversity at all of the sites, and they're labeled with the name of the river, between the diversity and several environmental measurements. And he found that one of the strongest relationships was with water travel time, particularly associated with the 4 largest river sites circled here in orange, the Sanga, Ubangi, Alima, and Congo rivers. So this supports our theory that river microbial communities are in constant flux, shifting from terrestrial communities that are dispersed into the river with groundwater into truly planktonic communities in the river channel itself. And all that's required for this to happen is time for the communities to grow and change. But what structures the microbiomes in all the smaller rivers and streams? We removed the 4 big river sites and reran the model and found that travel time was less important, right? Instead, we found a big gradient along the axis 1 in percent wetland cover in the upstream drainage area. And we circled those sites with greater than 50% wetland cover. So in small rivers where travel time is short and less important, landscape type and its control on river chemistry is more important in deciding the composition of river microbial communities. So our next step for this involves a taxonomic analysis of the communities to identify the dominant taxa that are in the big rivers and those that are associated with wetlands, greenhouse gases, and other variables. This is also exciting work from a natural history perspective because microbes in the Congo have not been described before, so this is the first study. So these results, they fit our model of controls on river microbial communities, which is adapted from the river continuum concept. At the landscape scale, This concept describes planktonic river microbial communities that require some time to develop, but are in a constant state of transition because they're constantly diluted with terrestrial organisms dispersed from land. So this study in the Congo suggests that there are many different terrestrial microbiomes, and they are linked to the landscape type, especially to wetlands. So good luck with the rest of the workshop. I hope you have a wonderful time and learn a lot, and I really wish that I was there. See ya.